Okay, wonderful. Hello, Daisy. It's lovely to meet you. We're very excited to see your presentation today.
Yes, I'm delighted. Should I go ahead and share screen then?
Absolutely.
All right. Hi, everybody. It's such a pleasure to be here. And thank you to the organizers for inviting me to come present and tell our story. My name is Daisy Robinton. I am the CEO and co-founder of Oviva Therapeutics. And I'm really thrilled to be able to share what we're doing today and tell you a little bit about our vision and mission and what we've been working on. If I can advance my slide. Here we go. So this may come as a surprise to some of you, but I hope not all of you. There has been a historic inequity that's existed in biomedical research and therapeutic development when it comes to gender science and understanding the sexes and physiology of females in particular. We know that biomedical research has been conducted primarily in male animals up until as recently as 2016, when the NIH, who's the largest funding body of biomedical research in the world, mandated that both sexes be included in all studies of grant recipients. As a result, the preclinical research that spans half of a decade has really skewed towards male physiology, including findings that describe diagnostic methodology, pathology, and therapeutic development. Now, this has been compounded by the fact that until the early 90s, inclusion of both sexes was not required in clinical trials in the US either. So this has led to poor outcomes for women across every facet of our healthcare system. So much so that between 1997 and 2000, 8 of the 10 drugs that were withdrawn from the US market elicit greater adverse events in women than in men. And it's not just an inclusion problem when we think about the biomedical research and the translation to the clinical paradigms. It's also a funding problem. Only 15% of our 2018 NIH budget was allocated to all of women's health. This includes conditions that are occurring in utero, prepubertal, fertility-related, pregnancy-related, pre- and postmenopausal, with only $43 million of that going to reproductive aging. Now, given that female reproductive physiology is one of the most robust multivariate signatures of human biology, it begs the question of why don't we do a better job looking more closely at this and understanding how we can integrate female physiology into how we think about women and patients and treatment options for them. So it's with this that we're renewing focus on female physiology and the ovaries at the heart of this that will bring women's health into the 21st century. And this is what Oviva Therapeutics is really an answer to. We're here to empower women with both confidence and agency over their long-term health choices around both fertility and aging, and really grounded in rigorous science and committed to securing an equal seat at the table for women's health. We'd really like to shine a spotlight on this connection between the ovaries and a woman's overall health and the aging process. And tangibly, we're doing this by developing first-in-class therapeutics that intend to provide choice for women to lead both longer and healthier lives. So how are we doing this? We have built our company on the premise that, or the fact that ovaries age asynchronously relative to the rest of the body. So really, they're the first organ to decline, and they do so at an accelerated rate. And you can see this depicted in the graph on the right-hand side here. We know that ovarian function rapidly declines after a woman reaches about 35 years of age. And by the time she hits roughly 50, they've essentially fully shut down, whereas the rest of the organ systems continue to function relatively well for several more decades. And this loss of ovarian function negatively impacts overall health, due in part to the systemic hormonal dysregulation that takes place when the ovaries decline. So with this menopausal transition that occurs as the ovaries decline in function, we see a whole host of systemic problems that occur in women. This includes an significantly increased risk of cardiovascular disease, declines in neurocognitive health, fatigue, sleep dysfunction, osteoporosis, sexual dysfunction, immune dysfunction, metabolic dysfunction, not to mention that somatically in the rest of the body, once you undergo the menopausal transition, we have evidence to suggest that there's an accelerated cellular aging that takes place in the rest of the organism. So really the ovaries and what they're doing are the seat of well-being for women. Now, this is also really interesting because there are some correlative data that suggest that ovarian health and function is linked to longevity, not just health and well-being. Women who go through menopause later tend to live longer lives, as do their male siblings. And women who enter menopause earlier tend to have shorter lifespans. Some direct experimental evidence has demonstrated that if we take younger ovaries from mice and transplant them into older individuals, we can extend health span by 11%. So while there is some indication that lifespan might be something that we could modulate here, I think really what's important to underscore and highlight is that we do know the ovaries are indelibly linked to health and well-being. And by improving their function, extending their function, we can extend the number of years of healthy and resilient women, lives that are available for years that are available for women's lives. Now, the way that we're approaching this is through something called the ovarian reserve. The ovarian reserve is a structure that dictates the timing of menopause, and really it represents the collection of eggs that a woman is born with. Each woman has an endowment of these eggs that decreases over time, and once it hits a certain low threshold, it introduces menopause and the ovaries fully decline. I think one thing that's interesting about this is that, you can see my cursor, we actually peak in our ovarian reserve when we're still in utero. So about 20 weeks gestation is when you have the greatest number of eggs. And this rapidly declines towards birth, where you have only 1 million left. And by the time you hit puberty, when you can actually use these cells, there's only 300,000 left. Now, what we're interested in here at Oviva is really this line here and the slope of this line, because once you hit puberty, you're losing about 1,000 eggs a month. No matter if you're on birth control or whatever else that might be happening, 1,000 eggs a month are being lost, with just 1 egg being ovulated each month. And so what we'd like to do is intervene here and extend the timeline during which this depletion occurs over. So if we zoom in a little bit closer to the ovary, I can explain in more detail how this works and then where we plan to intervene. So we're looking at the ovary here. It's this egg-shaped organ on the left, and the ovarian reserve represented here by these orange dots, which are the follicles, each one housing a single egg. Now, normally what happens is a group of this gets recruited from this dormant primordial phase into what's called a primary follicle. This then goes down a maturation. The follicles then go down this maturation pathway, with several of them undergoing degeneration called atresia, ultimately resulting in a single ovulatory follicle, which would release the egg for potential fertilization. And again, as I mentioned, about 1,000 of these follicles would be lost every month, just resulting in a single ovulatory follicle. And as I mentioned, we're born with an endowment of follicles that don't regenerate. So this is really our ovarian reserve, and it only goes in one direction, which is down. Now, how this is regulated is through a series of reproductive hormones. Most of you are probably familiar with the gonadotropins, FSH and LH. There's been plenty of therapeutic development to date around these hormones, both for birth control, contraception, and also for controlled ovarian stimulation and assisted reproductive technologies. But there has yet to be any development around this hormone called anti-Mullerian hormone. And what's really exciting and interesting about this is that it's the only known axis that can intervene at this early phase of follicle development. Now, AMH, as it's called, is produced and secreted from the maturing follicle. So once they've already been activated, the cells on the surface of the follicle secrete this as a means to regulate the pace at which activation occurs. So AMH helps control the rate of depletion of the ovarian reserve, which is why we're particularly interested in pursuing this as a therapeutic avenue. So Oviva is moving forward with a recombinant form of AMH as a means to modulate this ovarian reserve. And the way that we aim to move into the clinic and test this hypothesis in humans is by demonstrating proof of concept through improving fertility outcomes for women with diminished ovarian reserve. Now, as you can see in the graph, and as you may also know, women with diminished ovarian reserve have a very difficult time both achieving the appropriate number of eggs one might need from an IVF cycle to then lead to implantation and get a live birth, but also just to have a successful pregnancy through their process. And this can be due to a number of different etiological reasons, but a lot of it has to do with egg number and a lot of it has to do with egg quality. What we're looking at here, it shows you the number of cycles that a woman will go through and the proportion of those that will lead to a live birth, which you can see rapidly declines with age. Now, the type of protocol that they are exposed to for this treatment and for this paradigm is a standard IVF protocol, which includes both gonadotropins and GnRH antagonists. And with this conventional stimulation protocol, women with DOR frequently retrieve less than 3 oocytes. So their likelihood of having a successful pregnancy with this number is extremely low just at the beginning, which is why many of these women undergo multiple cycles of IVF, of controlled ovarian stimulation prior to attempting to implant. Now, what we're offering with our recombinant AMH therapeutic is a neoadjuvant treatment that would pose a block here, if you can see my cursor again, at this stage, preventing the activation of these follicles, providing a suppressive force. And while that might seem counterintuitive, what actually happens when you do that is that you synchronize the follicles that are ready to then be stimulated, so that when you remove the treatment, you have a greater than normal wave of follicles that are available for the stimulation, leading to a greater number of follicles that can be, or eggs that can be collected at the end of the stimulation cycle. What this has looked like in rodent models, following the same treatment paradigm, is that treatment with our recombinant form of AMH leads to a 3x improvement in egg yield without affecting quality negatively. And then when we take that into models of diminished ovarian reserve, so animals that have been treated with a chemotherapeutic agent that damages their ovarian reserve and approximates DOR in humans, we see still an improvement of 2x in terms of the number of eggs that can be captured with no impact on viability. So this strategy really represents a novel mechanism of action that would be a first-in-class therapeutic for this particular indication, but also first-in-class in terms of the axis that we're targeting and our ability to modulate the ovarian reserve without losing eggs, the way that some of the other treatment strategies will have happen. And just to bring it home and back to this graph, because we really are focused on the ovarian reserve as our approach to extend ovarian function, we really aim to change, as I said, the slope of this line, the pace of depletion that we're seeing, so that we can have this extension of health span between when one might naturally succumb to menopause and when they actually could have that pushed out due to a later date of slower depletion of the eggs, or potentially avoiding it altogether. And in some instances, you could even argue that we could enter a phase where we see women in healthy menopause. That is to say, they aren't cycling, they aren't having periods, but they aren't having the hormonal dysregulation that one experiences once you enter menopause naturally. And I think what I'd like to offer here is the thought that what we're trying to do is imagine a world where women have control over the menopausal transition, much like they were given control over their reproductive choices with the introduction of the oral contraceptive pill in the 1960s. That was a revolutionary advance for women that allowed them to control their physiology and their bodies in ways that, would be well to do to extend to other facets of how our reproductive cycle occurs over time. And with that, I would be remiss to not speak about our team, even just briefly. Especially my co-founders, Dr. Pat Donahoe and David Pepin. Pat was one of the first to clone and describe the AMH gene and its function early on, many decades ago. She's really built her career on the discovery around this. And David, who was formerly a trainee of hers and now has his own lab at Harvard MGH, he really described its role and function in the ovary specifically. He's an ovarian reproductive biologist. And having them on board with this has been really just, a dream and a pleasure and an honor, along with the rest of our team providing the R&D and operational support, BD support, and a special shout out to our scientific and clinical advisory team, because if we go back to the beginning of the presentation where I talked about this historic inequity that exists, somehow women's health has become this niche area, despite the fact that half of our population are women. And so finding the right expertise to understand the nuance of female physiology and help us to build out a preclinical and non-clinical plan that really captures that and takes into account all of the questions we need to ask and all of the things we need to think about. That's quite a challenge, and we've really gotten lucky with the folks that we have. They're quite experienced and have really brought our program to a wonderful place. So with that, I'll end there and allow some time for questions, and I'll turn it over to the hosts.
Thank you, Daisy.